contract mostly today. Oops. Don't forget from our last set of notes the hierarchy. In other words, the muscle, that's the tissue we're dealing with. It's composed of fascicles. Remember those bundles? And those bundles surround the individual fibers, the myofibrils. Each of those are made up of myofilaments. And then if we go even smaller within the myofilaments, we have two types, actin and myosin, which are going to be extremely important today. So how does this all start? Well, the muscular system is tied, like most systems, this time with the nervous system. So there's something called the neuromuscular junction, and that is a signal between the nerves and muscles. So what happens is a nerve impulse triggers something within the muscle. We'll get to that in a second specifically. But a big theme going forward with the rest of the systems we talk about is interactions and coordination between different organ systems is ridiculously essential. In other words, they're intertwined. They work together. We learn about them separately for the most part in here, but really they're intertwined here greatly. And today we'll focus on this, how the interaction of the nervous system ties in with the muscular system. So some vocab we're going to need. We already said the neuromuscular junction is where a nerve and muscle fiber come together. So you can see here, there is a little bit of space. It looks like it's touching, but it's not. This little space here is the neuromuscular junction. We have a motor end plate, the folded area where the muscle and neuron communicate. The synapse is that little gap between the neuron and the motor end plate. And synaptic vesicles. These are where neurotransmitters are stored. These are chemicals that trigger something, in this case a muscle contraction. So these synaptic vesicles hold neurotransmitters that'll get released into a synapse that'll go into the muscle and cause something to happen. So if we look at this picture, some of the key terms here. Number one is a neuron. So a nerve cell. We'll learn plenty about those when we get to the nervous system. Number two, well, we already know that. What surrounds muscle cells? Sarcolemma. So that's nothing new there. Three, a vesicle is really just something surrounded by a membrane that contains something. In this case, it's containing neurotransmitters. So these are synaptic vesicles. Four, the space between the neuron and the muscle cell. That is called the synapse. Five, hopefully you remember that shape from biology. That's the mitochondria. Lots of those in muscle cells because they make ATP, which the cell needs for energy. Six here, receptors. These are going to receive what the vesicles release into the synapse. And seven, these neurotransmitters specifically in the neuromuscular junction are going to be something called acetylcholine, or ACH. So go ahead, pause while you get all that into your notes. All right, so like we showed in the picture before, this kind of zooms in better. Here's our nerve cell, or our neuron. Here's the vesicles that contain the neurotransmitter, and we said that neurotransmitter is ACH, or acetylcholine. The acetylcholine is going to be what goes across and triggers the muscle cell to start its process of contraction. Here's our big picture stuff again before we get into the nitty-gritty. The muscle is our organ. A bundle of muscle fibers is our tissue. A single muscle fiber is a cell. And there's our levels of organization that we learned about really early in the year. So press pause here and kind of arrange them in a decreasing order. See if you remember this stuff. If not, you're going to need to know it eventually on the test. But in other words, what you would put first in this list would be the biggest structure, and then all the way down to the smallest. After you think you have it, unpause it, and we'll change the slide to show you the answers. Hopefully we said the muscle is the organ, the fibers are the tissue, an individual cell. Within that cell, we have myofibrils. Sarcomeres are those repeating units of myofibrils that are going to contract, and then those small filaments that make up the sarcomere are myosin and actin. So what's going to cause this? What's going to cause a contraction of a muscle? It's going to be the response to a nervous system function. In other words, the releasing of that neurotransmitter. And what we call the theory of how muscles contract is the sliding filament theory. Here's going to be some key players: ATP, calcium, myosin actin, acetylcholine. We will do some coloring on this so we get better at these terms in the next few days. So don't worry. I know we're throwing a lot at you, but we'll be good. Oops, sorry. So within, remember our organization, we said we have the myofibril. Within that, we have a sarcomere. And within that, we have myosin, which again are the thicker filaments, shown here in purple, and actin, which are thinner filaments, here shown in yellow. Why do we show that? To show the difference between a contracted and uncontracted or extended sarcomere. So look at this pictures. At the top here, this is a muscle that's relaxed. Remember, if you don't remember, go back here. Come on, go back. Myosin purple, actin yellow. So when we say that, we look here, actin purple, myosin yellow, and when the muscle starts to contract, you should see some differences. This Z line holding the myosins has moved. So has this. The myosins themselves are getting closer towards each other as the muscle contracts. And then a fully contracted muscle, it extends that even more. So our Z lines where the myosins are get pulled in, and so do our myosins. Why? Well, we'll get into that here in a second, after you put this into your notes. So let's take this down to a molecular level. And this video is going to demonstrate some things, and then we'll put some notes from it in our note guide, and I might pause a few times to explain this, but this video is short, about a little over 2 minutes, but it's better than just hearing it, so here we go.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments.
All right, so they said what triggers the muscle contraction in the first place, a nerve impulse comes through. That releases calcium. Who cares, right? What's calcium got to do with this? No, calcium's an important player. It says calcium is going to bind on the thin actin filament at this site called troponin. Once it does that, it's going to change the shape and open up an area that myosin can join and attach to. They'll show that here now.
Are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites.
So, without calcium, the actin will not turn, and it will not be able to accept the myosin, which is necessary for it to contract.
At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule.
Remember from biology, ADP means two phosphates joined to something called adenosine, and here's the third phosphate. So these come from ATP. When we talk about energy in the cell, here's what we're talking about.
Remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin.
When they release the phosphate, they're using it for energy, which allows them to move and bind to the actin.
Myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the
Notice the ADP fell off because it was used. The energy was used to this motion of pulling the actin filament.
Myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and...
So the contraction stops when ATP comes in, joins the myosin heads. That causes them to release from the actin, thus not being able to pull it and contract anymore.
An actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
All right, hopefully that lets us visualize this a little better. Go ahead and pause while you put in the notes from the video. So like the video said, we're going to kind of break it down now in different diagrams, but this is all referring to stuff we saw in the video. The action potential is simply the nerve impulse. So a nerve is getting an impulse. We'll talk about how those work when we get to the nervous system. And it causes, oh, look at that, one of the words we talked about earlier, this unit, sarcoplasmic reticulum, which stores calcium to release calcium. Why do we need calcium? Well, remember, like the video said, calcium binds on troponin. When it does that, it creates a change. It rotates the tropomyosin, this gray rope, and it exposes the myosin binding site. So without calcium, the shape of the actin doesn't allow it to join with myosin. Once calcium binds to the troponin, it turns the tropomyosin around, exposing the place, the binding site, where the myosin head can come in and attach. Where does it get the energy to do all this? Remember, ATP, cellular respiration is the process that makes that. With taking in oxygen is necessary for that. This is just a quick review slide from old biology stuff. So, big picture here. I'm going to break it down into each of these slides separately, but remember where we start. The purple myosin and the thin yellow actin. And then we're zooming in to watch that place that they interact. So, remember, as we have it now, we have ATP with the myosin head, so it doesn't have enough energy to do much. Once we break off a phosphate, now we're using that for energy, we have ADP, which means two phosphates, diphosphate, and a separate phosphate. Well, now we have enough energy, we can do something. So as long as calcium came along and helped change the shape of this actin, myosin can now join and form a bridge where it attaches to the actin, pulls it, shortening it, and when it does that, it takes energy, so it releases the ADP and the phosphate and pulls it. More ATP comes in, causing it to release, and sets us up to repeat this as long as we have calcium and ATP present. So we break it down into each step. Here's the zoomed out version. Remember, purple, thick, myosin, actin. It cannot bind unless we get some calcium, and the calcium won't get there unless we have a nerve impulse. Remember, the nerve impulse triggers the release of calcium, and then once calcium comes, it's able to, actin is able to be bound by myosin as long as myosin has enough energy. Where does it get that energy? From ATP by releasing a phosphate and turning to ADP, as we show here. Once we have that, we're going to be able to now bind and form a cross bridge, and we're going to use the energy we have present in the ADP and phosphate to pull that actin, causing it to get shorter and contracting the muscle. That's all it is. Well, when we do that, we release that source of energy. We used it up, ADP and phosphate, and we've pulled it. Now, to get back where we started, we just need more ATP to put the myosin head back in that low energy state. There's a kind of quick picture there, or a quick little diagram GIF, and you can see what's happening. You see these yellow chemicals coming. So that's going to be our calcium, because when it does, it turns the troponin, turns the blue tropomyosin, allows this, which is the myosin head, to bind. It pulls it, and notice these ATP is coming in, changing to ADP and phosphate, coming off. So really, this is a neat little short repetitive GIF, but it shows everything we've talked about. If you want, you can open up your own slideshow and click this link. It's pretty neat. We're not going to go over it here, but it lets you explore if you need to learn a little more about what we're talking about. All right, so muscle and exercise. When you exercise, your muscles get tired, they fatigue, and when they do, they lose the ability to contract if you use them long enough. In other words, no matter how good a shape you are, if you went running forever, eventually you're going to run out of energy and not be able to go. A muscle cramp is a sustained involuntary contraction. So your muscle keeps contracting when you don't want to, and they can be very painful. When you don't have enough oxygen, because you're exercising so much, you're not able to produce enough ATP. It's called oxygen debt, and that causes an accumulation of lactic acid. It's a type of fermentation that occurs when you don't have enough oxygen. And it does make some extra energy, some extra ATP, but it also makes this thing called lactic acid, which makes you sore. We're not going to watch this one together, but I'd recommend you going to YouTube and Googling or YouTubing the surprise reason our muscles get tired. It's worth watching, but won't be on the test. So changes in muscle, both good and bad. Hypertrophy. If you lift weights to get stronger and bigger, that's what you're trying to do. You're trying to make your muscles get enlarged. So it can be a good thing. Atrophy, though, is when you don't use your muscles and they become very small and weak. So often people who are paralyzed, like from the waist down, well, their legs atrophy. Their leg muscles get really small because they don't use them. People who go in the space station for long enough actually have some atrophied muscles, and they have to get back into, build those up when they get back to Earth. Come on, why is it not changing? All right, let me see what's going on here. Oh, because I forgot to put those in. So sorry about that. Let me open something up real quick so you get the last part of notes, and that's a bad job out of me here. But I should be able to find it relatively quickly and let you get the last part of the notes you need. There you go. Go ahead, pause this as long as you need it to copy the last of the notes, and then you are done.